Hi everyone, welcome to the HPAI workshop for this meeting. Today I'm going to start you off with a little bit of avian influenza 101 and provide you with some context of the outbreaks occurring in seabirds. So it's no doubt that as you've been learning about this virus and reading about this virus, you've come across lots of really complicated names. And so where I wanted to start today was just to try to decode and demystify this complex name for these viruses. And I'm going to start here right at the end on this part called H5N1. So on the right here, we have a picture of avian influenza viruses. And on the outside, you can see two different color proteins sticking out. This is called the HA and the NA. Now, these proteins are highly diverse and have been subdivided into H1 to H16 for the HA and N1 to N9 for the NA. These viruses are encoded on different parts of the genetic material of the virus, which means that the virus has the capability to mix and match these. So we can have H5N1 as a combination, we can have H7N7 as a combination. Pretty much every combination that you can imagine has been detected in nature to date. Avian influenza viruses are divided into these two broad categories based on their pathogenicity. So we have low pathogenic avian influenza and high pathogenic avian influenza. So low pathogenic avian influenza, sometimes called LPAI, comprises all the HA subtypes found in birds, so H1 to H16. It's common in wild birds, particularly members of the Anseriformes and Pteridroformes, and is believed to be a natural part of the virus communities of these birds. These viruses cause no disease in their wild bird hosts, no death in their wild bird hosts, but we may see occasional mild disease in poultry. This is in contrast to highly pathogenic avian influenza viruses, which is what we're discussing today. These viruses comprise only the H5 and H7 subtypes. They have caused outbreaks in poultry and are associated with disease and death, both in poultry and in wild birds. Now, low-path and high-path viruses are evolutionarily linked, and I've sort of explained that in a little diagram here on the right. So what we have is these low pathogenic H5 or H7 viruses that are found in wild birds. It's a natural part of their virus communities. These viruses enter poultry production, and we see really significant and substantial amplification of these viruses, which may lead to genetic changes in the HA, which sort of results in these highly pathogenic H5 or H7 viruses. Essentially, the viruses go from viruses that replicate just in the gastrointestinal tracts of wild bird or respiratory tracts of poultry to viruses that can replicate anywhere in a bird. And this is what causes the sickness, disease, and death in birds. Now, the last part of that name is that 2.3.4.4b bit, and that's a little complicated, but it essentially describes the sublineages of H5. Okay, so remember we have HA and NA. Within the HA, we have H1 to H16, and H5 being one of those. And then within H5, we have lots of genetic subdivisions. And so how I wanted to explain this was through this diagram here, which is a phylogenetic tree. So it's just a tree of relationships, and it's scaled here by time on the bottom. So on the top here in the blue, we have lineages of low pathogenic H5 viruses that are found in wild birds. Here, starting in red, this represents the first detection of this lineage called the GSGD, which you may have seen. It's called the Goose Guangdong lineage. So this was the first detection of this highly pathogenic H5 lineage. Through time, this lineage has evolved and diversified, which you can see here in this tree, sort of through these little subclusters. And over time, we've tried to find a way to track these clusters and give them names that are useful to us. And so what has resulted are these really long number and sub-number schemes. So 2.1.3 and 2.2.1, for example. In 2014, we saw the evolution of this clade called 2.3.4.4, and it spread quite rapidly over the globe. Now, 2.3.4.4 has evolved since into sublineages, A, B, C, for example. And so what we're talking about is clade 2.3.4.4b. Now, that bit, I'll just remind you, refers to the HA part, the H5 part, okay? So high path refers to genetic changes in the HA, and the 2.3.4.4b part is the sublineage of the HA. But we have this NA thing that can mix and match. So for the first 20 odd years that this lineage was detected, it was always detected with an N1 subtype. With the evolution of 2.3.4.4, we saw different NA types, which is why it's sometimes written as H5NX. We had lots of N8s and N6. And what we saw starting late last year was the return of this N1 coming back. But it's not the same thing as the old H5N1s because we have a different HA lineage that we're discussing. Now, changing tact, I just want to talk a little bit about avian influenza viruses in seabirds. Now, prior to the 1960s, avian influenza virus had only been described in poultry. But then there was a mass mortality event in South Africa in common terns, and an H5N3 virus was isolated. So this allowed us to appreciate that these viruses are found in both poultry and in wild birds. About 10 years later, we detected avian influenza virus in healthy birds, in this case in wedge-tailed shearwaters in Australia. And this really led us to appreciate the enormous diversity of low pathogenic avian influenza viruses, and that wild birds are a reservoir for these viruses. As time has continued, we've been sampling lots of different birds and appreciate that members of the Anseriformes and Trageriformes are really the central reservoirs for these viruses, but seabirds are also implicated in the ecology of these viruses. We did a review a few years ago, and this is a figure from the review, wherein we identified 4 groups of seabirds that are likely important in the ecology of these viruses, including penguins, terns, Persillariformes, and Elcidae. And how this figure works is it's just showing you sampling effort over time. Showed here is a heat map, so year on top, different tissues, and then isolates. Okay, this is the number of viruses that we've been managed to isolate from these species. In addition to these 4 groups, we know that gulls are important reservoirs for these viruses. In fact, they are the main host of H13 and H16, which are currently only found in gulls and terns. Now, I'm not going to discuss the highly pathogenic outbreaks in seabirds that are occurring in the Northern Hemisphere, as there are many speakers following me that will talk about this in detail. I just want to mention that while the highly pathogenic avian influenza outbreaks in seabirds that are occurring in the Northern Hemisphere are unprecedented, this virus has been a scourge to Southern Africa for a number of years now, causing many outbreaks in various seabird species. And I expect that there are probably a number of really useful lessons that we can learn for the outbreaks in the Southern Hemisphere and apply that to our response here in the Northern Hemisphere. Now, just to wrap up, I just wanted to mention that we are organizing ourselves a little bit better, and we have started a consortium for highly pathogenic avian influenza viruses in seabirds. Our consortium currently comprises experts in avian influenza in wild birds, seabird ecologists, people on the ground responding to outbreaks, as well as decision makers. And one of our aims is to try to provide an appreciation for the scale of the outbreaks that are occurring globally. And so we will be doing some work in the next weeks and months to try to collate a lot of the outbreak data that many of you will have been providing through the notification system to the OIE, which is now called WOA. And you may also have seen this survey circulating on Twitter, which is also our doing to try to provide us with more information about what is going on. And lastly, I just wanted to provide you with some resources, some good papers that are hopefully useful to you. Thanks very much.